will see about synthetic seeds or artificial seeds. Synthetic or artificial seed technology involves the production of tissue culture derived somatic embryos, which is encapsulated in a protective coating. Synthetic seed is confused with synthetic cultivar. The synthetic cultivar is defined as an advanced generation of an open pollinated population composed of a group of selected inbred clones or hybrids. The need for artificial or synthetic seed production technology. In some crop species, seed propagation has not been successful for the following reasons: heterozygosity of seeds, minute seed size, presence of reduced endosperm, requirement of seed with mycorrhizal fungi association for germination, such as orchids, and seedless varieties like grapes, watermelon, etc. In those crops, synthetic seed or artificial seed will be an effective solution for mass multiplication. And artificial seed production technology is currently considered as an effective and efficient alternate for the conventional techniques. The advantages of artificial seeds: it is easy to handle, store and transport the artificial seeds, and long-term storage with maintaining the viability is possible, and it maintains clonal purity and uniformity, and mass propagation in economic way is possible. Artificial seed creates new genotypes. Now we will compare the synthetic seeds with existing propagation techniques. First one is micropropagation. Produce medium scale multiplication of propagules. Since initial establishment costs are high, high cost per plant is incurred, and moderate or low multiplication rate is observed here, and acclimatization is required. Second one is greenhouse propagation. Here we can multiply only in small scale level, and high cost per plant is incurred, and it is limited to mother plant. The number of propagules less or high depends on the nature of mother plants, and mostly rooting is required with certain kind of hormones. Whereas in the case of synthetic seeds, we can produce large scale, and comparing total cost, low cost per plant is required, and rapidly we can multiply, and directly we can plant the seeds. The basic requirements for production of artificial seeds are production of high quality vigorous somatic embryos. We can encapsulate with certain gelling and complexing agents so that we will get the artificial seeds. The steps in artificial seed production. So initially we collect the explant and produce the somatic embryo in a sufficient numbers. We can mix this in a solution containing gelling agent. Most commonly sodium alginate is used. Later another compound called complexing agent is added drop by drop. So the complexing agent reacts with the gelling agent and it will form the capsule-like structure. Somatic embryo already mixed and optimized in a way that each capsule contains one somatic embryo. Then we have to test the encapsulated seed, and we can store this kind of artificial seeds. Whenever we need, we can send it for germination. Now we will see the gelling and complexing agents used to produce the artificial seeds. The common gelling agent is sodium alginate 0.5 to 5%, sodium alginate 2% with gelatin 5%, carrageenan 0.2 to 0.8%, locust beam 0.4 to 1%, gel rate 0.25%, and other agents such as agar, polyco, carboxymethyl cellulose, guar gum, sodium pectate, track and gum also reported. Compared to all of this, alginate encapsulation was found to be more suitable and best one. Next is complexing agent. Calcium salt up to 30 to 100 micromolar. More preferably calcium chloride 30 to 100 micromolar is used. Potassium chloride, ammonium chloride also used. If ammonium chloride is used, temperature is lowered for best encapsulation. Generally, three kinds of artificial seeds are produced. One is hydrated artificial seeds. This is reported by Redenberg et al. In 1986. Here the mixing of somatic embryos with sodium alginate solutions followed by dropping of the complexing agent calcium chloride is added drop by drop. The beads become hardened as calcium alginate is formed. In general, a waxy coating is provided over the beads to prevent the water loss. And next type of seeds are called desiccated artificial seeds. This is proposed by Kim and Janik in the year 1989. Here the synthetic seed coats to clumps of somatic embryos to develop the desiccated artificial seeds. Here equal volume of embryo suspensions and 5% solution of polyethylene oxide, popularly known as polyox, is added. Then it is subsequently dried to form the polyembryonic desiccated vapors. And these vapors are subjected to hardening treatments with 12% sucrose or 1 microliter of abscisic acid, followed by chilling at high inoculum density. The third type of artificial seed is called fluid drilling somatic embryo seeds. This is proposed by Gray in 1987. Somatic embryo suspended in a viscous carrier gel which extrudes into the soil. The key problem is sucrose is major ingredient added in this technique, which is subjected to rapid growth of microbial contaminations. Now we will see the alginate encapsulation mechanism with the matrix. The alginate is a straight chain hydrophilic colloidal polyuranic acid present in the brown algae used for the food stabilizer. When somatic embryo is mixed with sodium alginate solution and complexing agents such as calcium chloride, ion exchange between sodium and calcium will cross-link and form rounded and firm beads. The hardness and rigidity of the capsule mainly depends upon the number of sodium ions exchanged with calcium ions. In general, 3% sodium alginate solution with 75 millimolar calcium chloride gives optimum bead hardness and rigidity. The volume of sodium alginate is optimized to contain 1 somatic embryo per droplet. Next one is artificial endosperm. The addition of nutrients and growth regulators and additional substances to the encapsulation matrix is desired, which serve as an artificial endosperm, supplying the nutrients to the embryo. This increases the efficiency of germination and viability of the encapsulated somatic embryos. In addition, some adjuvants also added to the matrix, such as nutrients, carbon source, growth regulators, fungicides, pesticides, antibiotics, microorganisms added. These agents prevent the embryo from desiccation and mechanical injury. Activated charcoal also added for the two benefits. One is it breaks up the alginate and thus increase the respiration. Second one, it retains nutrients within the hydrogel capsule and slowly releases them. So now we will see the synthetic seed production with different propagules as a material. Somatic embryos are widely used to produce artificial seed in crops like alfalfa, celery, brinjal, carrot, brassica, lettuce, potato, sweet potato, cucumber, asparagus, rice, horseradish, wheat, triticale, maize, sorghum, soybean, sugarcane, coffee, tobacco and cotton. Whereas auxiliary or adventitious buds are used for the and demonstrated for the production of artificial seeds in mulberry, eucalyptus and grapes. Shoot tips have been demonstrated for the production of synthetic seeds in banana, cardamom and carom carvi. Utilization of artificial seeds. So the multiplication of non-seed producing plants is possible, such as ornamental hybrids, polyploids with elytrites, and propagation of male or female sterile plants for hybrid seed production is achieved through synthetic seed production. And germplasm preservation is possible, particularly in recalcitrant species such as mango, cocoa and coconut. And we can maintain the transgenic plant using this technique, and a single gene can be inserted into a somatic cell, and artificial seeds produced in tissue culture are free of pathogen. Feasibility of artificial seed production technology. In general, synthetic seed of the seedless watermelon would actually cost less than the conventional seed and provide a benefit at the outset of crop production. We have to consider it may either reduce production cost or increase the crop value, whether its use is justified for the given crop species. Thank you.